Good evening, everyone. My name is Damaris. I'm a toxicologist working in the Department of Clinical Medicine. I primarily work with the health effects of microplastics during pregnancy. So before going on to my project, I would like to give a brief introduction of microplastics. So by definition, they are plastic particles less than 5 millimeter in size. They are considered a unique contaminant of concern because of their dual toxicity. That is, they have particle toxicity and chemical toxicity. Most of the plastics we know have inherent chemical additives. Along with that, they can also act as carriers of different other environmental contaminants like pesticides, heavy metals, and pathogens. So as we know, we are constantly exposed to these contaminants through two main routes, the inhalation and ingestion route. And over the past few years, there have been a lot of research going on about the amount of microplastics in different food and beverages, of which seafood is one of the most best investigated group in which microplastics have been detected. And not surprisingly, there have been also reports of microplastics in blood, liver, placenta, lung, kidney, fecal samples, etc. However, the potential health effects of microplastics have not been very well characterized, and there is insufficient data, especially in vulnerable groups such as pregnant women. So to bridge some of these knowledge gaps, we have developed this project in which we are dealing with two main research questions. That is, can microplastics from maternal diet reach the fetal environment, and whether mother's plastic exposure has an effect on the fetus. So this experiment has two major parts. One is an animal study, and another one is a clinical study. In the animal study, we conducted a feeding trial for 18 days. That's the normal gestation period of a pregnant mice. So we have three different groups, 8 animals each. One is a control group, which received normal feed without any microplastics. Then we had a high exposure group in which they received feed spiked with microplastics at a dose of 0.5%, and a low MP group, which received feed with microplastics at 0.1%. And at the end of the trial, these animals were euthanized, and placenta and fetal brain were collected. The RNA was extracted and was sent for RNA sequencing to study the differential gene expression. For the clinical study, this was a human observational study in which we recruited 30 pregnant women in their 3rd trimester. We did a comprehensive dietary assessment. Then we did an extra ultrasound for them to study the blood flow in the fetal body. We collected samples of placenta and cord blood during delivery, and also the fecal samples before delivery from the mother. And the samples are being processed, and the microplastics are being characterized in these samples. So coming to some of the preliminary findings, from the animal study, it was found that the fetal brain was more susceptible to microplastic exposure than the placenta. In the placenta, there was certain immune-related genes which were upregulated. That shows that they have an immune response. The placenta have an immune response to the ingestion of microplastics. And in the fetal brain, several genes related to neurodevelopment neurodevelopment pathways were altered. That means the microplastics have a neurotoxic effect on the fetal brain. There was also clear dose-response effect. That means the high MP group had significantly higher expression of genes than the low MP group. Coming to the clinical study, this is an ongoing study, so I don't have all the results. We have analyzed half of the fecal samples, among which about 70% of the participants had microplastics in their fecal sample. The particles consisted of commonly used plastics like polypropylene, polyethylene, polystyrene, and PET. There was no significant difference. So far, the participants we have analyzed, there was no significant difference in the microplastic abundance between the high and low seafood consumers. However, there was a positive correlation with their plastic use, but this is not very conclusive because we have still more data coming from the rest of the participants. So to conclude, as for the evidence from the biological effects of animal study and also the widespread microplastic exposure in women among late pregnancy, it is necessary to have more long-term longitudinal research, which is quite lacking in this research field. And also, even though the understanding impacts of microplastic is quite scientifically complex, but it's urgent because even subtle disruptions in fetal development can have long-term, long-lasting effects. That's all I have for you today. Thank you.